Good morning everyone. So today I'm going to discuss the experiment number 1 as given in your course to study the pollen grain germination on slide. In this experiment we will study the formation of pollen tube. So how it take place and what sample we are using in this experiment. Let us discuss about the procedure in detail. In this experiment we take sample as hibiscus flower as I have shown in the video. The hibiscus flower we take stamens part of it, and stamen, as you know, this structure is stamen. It is having a stalk filament and a bilobed anther on it. As the anther contains pollen grains and we have to observe the pollen grain germination on slide, so we are extracting the pollen grains from the anther. These pollen grains we put on the cavity slide. I have shown diagrammatically and also I have shown in the video also. The cavity slide has cavity or space on it on which you can put the sample. We dust the pollen grains, we take it out from the anther, we dehisce the anther first, we take out the pollen grains and we dust on the cavity slide. Now what will be the next step? We have to add 10% sugar solution onto this slide after dusting the pollen grains. This 10% sugar solution, as I have explained, that it contains sucrose, 10 gram of sucrose. Beside it, it contains calcium salts, potassium salts, and you can also add boric acid in it because it enhances the pollen tube formation. Then the next step we do is, after putting the sugar solution, after adding the sugar solution, we stain it with the safranin stain so that we can highlight the pollen grains on slide. And then we have to put cover slip on it. After putting the cover slip, we have to observe it under low magnification, under compound microscope. There you can see the pollen tube formation. Now how will it look like? Let us see. When you observe the slide after 5 minutes, then you will observe that this structure, we call it as pollen grain. It can be seen on the slide very clearly. It is having germ pore, this particular space which doesn't contain exine. As you know, there are two layers of pollen grain, exine and intine. Right? And the pollen tube formation take place through this particular pore. We call it as germ pore. Whenever